all that is missing in prokaryotes. In prokaryotes, there is no nucleus, there are no membrane-bound organelles, right? So there is no regulation at the level of post-transcriptional processing or pre-transcriptional processing and even post-translational modifications to that extent. So all that regulation if you want to study, all that processes if you want to study, it is not possible in case of prokaryotes, right? For that you need to move to eukaryotes. But then if you move to eukaryotes, we know most of the eukaryotes are multicellular. Then we do not have those advantages. It cannot give you a clonal population of cells, right? Except for the zygote which can grow to give you an embryo, right? And it is not ethical to work with stem cells, right? So we are stuck again, and again haploidy is not there. So that dominance resistance will come now. You got my point? So the biggest advantage of working with yeast is that it is both unicellular and it is both eukaryotic, right? And then yeast has two forms. It has haploid form as well as a diploid form. It can grow in both the stages. You must have studied in your school level that yeast. If you've done microbiology, then also you must have studied that yeast can undergo both sexual reproduction as well as asexual reproduction. So what is the asexual mode of reproduction for yeast, for Saccharomyces cerevisiae? How does it grow itself? That is, its name is budding. It's called budding yeast, right? And you have another form of yeast which is the same genus but a different species, that is Saccharomyces pombe. So pombe is the fission yeast. So that divides by multiple fission. Multiple fission is another mode of asexual reproduction, like budding. Budding yeast divides by budding, and fission yeast divides by multiple fission. Right? So budding yeast name is Saccharomyces cerevisiae, and the fission yeast name is Saccharomyces pombe. Pombe is P-O-M-B-E. Pombe. So just the species name is different. The genus is the same, Saccharomyces. Right? So it has asexual mode of reproduction. And yeast can also have a sexual mode of reproduction when it forms a diploid stage. Now you would understand that without having a diploid stage, sexual reproduction cannot happen. Why is that so? Yeah. Yeah. And what form of cell division usually takes place in sexual reproduction? Is it mitosis or is it meiosis? Meiosis. So meiosis requires a diploid cell, or it can happen in a haploid cell also. You need a diploid cell because it is reductional division. You cannot assume that it has, say, 5 chromosomes and they get halved. And it has 2.5 chromosomes. It's not possible. It has to have 5 pairs of chromosomes. So the one pair goes to one cell and another pair goes to another cell. So only a diploid cell can undergo meiosis, right? So that is why it is a real advantage to work with an organism which can have both the kind of reproductions, asexual as well as sexual. So you can study both the things, right? Now we were doing homologous recombination. So can homologous recombination make any meaning in an organism having haploid genome? Again, no, right? Because homologous recombination means that you have 2 copies that are homologous to each other, which means you're already talking of a pair of chromosomes, right? So again, we are talking of diploid level. So Saccharomyces cerevisiae can exist in many different cell types. So these are cell types or mating types. So they are having almost similar genome, but there's slight differences. You could call them as kind of strains. So the genus is the same, the species is the same, but the strain is different. For example, if you say coronavirus, this particular strain, coronavirus, that particular strain, the genus remains the same, the species remains the same, the strain is different. So here also you have certain mating types in the yeast. So they are kind of strains which are slightly different in the genomes, very, very slightly different. It could be just one single locus or one particular region of the genome which might be different, otherwise it is all similar. So there are different mating types like A and alpha. So when it is A type, it is haploid. When it is alpha type, it is haploid. But when there is a mating between A type and alpha type, then it forms a A alpha genotype. So you can imagine that when it is A, it is haploid. When it is alpha, it is haploid. But when it is A alpha, it is diploid. Right? So it's a mating between A and alpha types. So they can grow in both haploid state and diploid state. This I've already talked about. So if you look look at the life cycle of Saccharomyces cerevisiae, there are genome, but there's slight differences. You could call them as kind of strains. So the genus is the same, the species is the same, but the strain is the different. For example, if you say coronavirus, this particular strain, coronavirus, that particular strain, the genus remains the same, the species remains the same, the strain is different. So here also you have certain mating types in the yeast. So they are kind of strains which are slightly different in the genome, very, very slightly different. Could be just one single locus or one particular region of the genome which might be different, otherwise it is all similar. So there are different mating types like A and alpha. So when it is A type, it is haploid. When it is alpha type, it is haploid. But when there is a mating between A type and alpha type, then it forms a A alpha genotype. So you can imagine that when it is A, it is haploid. When it is alpha, it is haploid. But when it is A alpha, it is diploid. Right? So it's a mating between A and alpha type. So they can grow in both haploid state and diploid state. This I've already talked about. So if you look at the life cycle of Saccharomyces cerevisiae, there are, sorry, this is not italicized. Please make sure whenever you write down any scientific name, it has to be italicized. Or if you're writing with your hand, it has to be underlined. And underlining should not be a common underlining for both the names. The genus has to be underlined separately, the species has to be underlined separately, which means the line doesn't connect. Right? So Saccharomyces underline, then cerevisiae underline. So life cycle of Saccharomyces cerevisiae. So there are two haploid cell types which are kind of spores. So yeast has spore form. So it can have haploid spores. So A and alpha type. So if you have an A form, it is going to form A type of spores. If it is alpha form, it is going to form alpha type of spore. And that's the kind of vegetative reproduction which is asexual, which means it is just dividing by budding. So yeast will give you more of A spores via budding. Right? Now there is conversion. There is a conversion possible between the haploid and the diploid states, which is mediated by mating and spore. So mating means, for example, if there is a mating between A and alpha, it can form A alpha. This is a kind of state which can be formed. So haploid can become diploid. Now when A and alpha cells grow together, these cells make to form A alpha diploid cells. And under the conditions of reduced nutrients, these diploids undergo meiotic division. So it is basically the stress condition where you have reduced nutrients available for the yeast that your A alpha cells can undergo meiotic division. So the sexual mode of reproduction begins only beyond stress conditions. Otherwise, it is mainly vegetative. Vegetative means asexual, which means budding. Right? Vegetative versus the sporulation. Now, the mating type genes encode transcription regulators at a particular locus known as MAT locus, and MAT locus stands for mating type. MA is from mating, T is from type. So MAT locus is mating type locus. So here there are some transcription regulators and they define the genotype of that yeast, whether this yeast will be A type or it will be alpha type. Now what we are doing in this topic is mating type switching. So it is possible that a yeast switches from one form to another form, which means it can switch from an A form to alpha form. So that is what we are trying to understand. How does it happen and if it involves homologous recombination, because that is why we are doing this topic here. Yeah, so when you have reduced nutrient conditions, it is generally the signal for the sporulation to happen. So when there is sporulation, you have formation of both A and alpha, and they tend to fuse with each other, and that is how when an A spore tends to fuse with the alpha spore, it forms an A alpha diploid. So this A alpha diploid, when it wants to grow itself, the only way possible is generally the sexual reproduction. Yes, because sporulation is something which generally happens during reduced less availability of nutrient. Because sporulation means that the cell is trying to cover itself inside a coat and maybe remain as dormant. So when nutrient is not available, that makes sense, because it doesn't want to spend time on growing right now. When it is mitosis, then it is just A alpha making A alpha. Right? But when it is meiosis, it is going to form both, it is going to form two A's and two alphas. What we are discussing now is how does an A form itself switch to alpha form? It's a very unique phenomenon, and it is found only in case of yeast. It is not seen in other organisms, right? And it involves homologous. So in A cells, the A1 gene is present at the mat locus. In alpha cells, we have alpha genes, and there are two alpha genes, alpha 1 and alpha 2, at the mat locus. So what you need to understand here is that in the mat locus, if you have A gene, it is going to be A strain. If in the mat locus it is alpha gene, it is going to be alpha strain. I'm just going to show you the figure. And the cells can switch their mating type by recombination. What is the most important lesson that here you need to understand is that the fact that cells can switch their mating type via homologous recombination. I'll skip this part. You can come back to this. So just look at this figure. You do not need to go into too much of detail on mating type switching. Just understanding the phenomena, how does it happen, briefly is enough for you. You don't need to remember the steps and all. Just understand the concept that how it is possible using homologous. Right? I'm not really going in detail. So this is your mat locus. Mat locus is the mating type locus. I told that whatever gene this mat locus contains, that is the genotype of that strain. So for this strain, you have A here. It says mat A. So it has the gene A1 inside. So this is the A cell, right? Do not look here, do not look here, just look at the mat locus. If it is mat A, the strain is A. If the locus has mat alpha, the strain is alpha. Is it clear till now? That whatever gene is there in the mat locus, that is the genotype of the organism, because that is what is getting expressed. Then what you see on the left hand side and right hand side of the mat locus is a silent locus here and another silent locus here, which means it is not expressed. What is expressed only is the mat, right? So there is this left one and a right one, HML alpha, HMRA. So HM stands for homothelic mating type. This is written here. HML is homothelic mating left, and HMR is homothelic mating right. Homothelic, so you know that hyphae and thellus, they are synonyms. So he is from hyphae. So homothellus, when you have of the similar type, then it is known as homothelic. So HML is homothelic mating left. So there is one silent locus on the left of mat locus, and there is one silent locus on the right of the mat locus. So this is the left one, this is the right one. So in case of A cell, you see a HML alpha here and a HMRA here. Similarly, you see the same types here also, right? Now, so whether it is A cell or alpha cell, you have this part and this part common in both the cells. The only difference is what lies in the mat locus, and that makes the strain different from the other one, right? So how does the switching happen? The switching happens via a looping, where there is a homologous recombination between the HM locus and the mat locus. Now, if the switching happens, if the homologous recombination happens between HML alpha and mat A, then this A here gets replaced by alpha. So it becomes an alpha cell. Because in homologous recombination, you have an exchange, right? So the exchange is happening between homologous regions, and there is homology in the region which is having this locus as well as both these locuses. Because you have alpha here, you have A here. Here also you have A. So if an A and alpha genes are also not very dissimilar, they are more or less similar. They are very much homologous. So it is possible that homologous recombination can happen. So imagine that this goes and loops out here, right? So if it is like this, it might loop out like this, and then you have regions which are similar here. Because A and alpha also have homology. If it is A and A, it will not make any difference. If one is A, another is alpha, there could be a strand exchange, and what you get at the end is A here and alpha here. Now this was mat locus. This was HML. So what comes in the mat locus? alpha. So what is the genotype of this one? The lower one. Because MAT is the one that is expressed. The left and right ones are not expressed. So whatever comes in the center, that is a genotype. Right? So if the homologous recombination happens between HML alpha and MAT A, so A gets switched with alpha, that is why A cell forms alpha cell. That is why you see this arrow here. A cell gets converted to the alpha cell. Similarly, if you have a homologous recombination between HMR A, which is again a silent locus, and MAT alpha, so when A comes here, alpha goes here. So when A comes here, it becomes S, because that is the only dissimilarity between the two. Right? So A can switch to alpha, alpha can switch to A. Yeah. So it is happening in one chromosome because it is haploid, right? It is just a looping in mechanism in the same molecule, using the same molecule, because there are homologous regions in the same genome. So now, because we're going to begin with another topic, that is site-specific recombination, there also you will observe that within the same molecule you can have homologous recombination if there are homologous regions, just by looping in and other things. And that is how all the genetic rearrangement happens. That is how your case can switch from one meeting type to another. Okay? Just coming back to this thing. So just go through it. You don't need to really go in detail. Nobody is going to ask you the process of mating type switching. Just understand it, that you could be simply asked a simple figure and explain that what is happening, that how does homologous recombination happen. So this is what I've already explained, that when recombination occurs between MAT and HML, when A cells switch to alpha cells, and similarly the other way around. So when it is HML, then A switch to alpha. When it is HMR, then alpha switch to A. The same figure that we did here. Because here it is happening between HML and this, and here it is happening between HMR and MAT. And how does it happen? So it happens the way homologous recombination happens. In homologous recombination, you always need to have a strand break. Then you need to have recession of the or the exonuclease dilation of the 5' or 3' ends, and then there is filling up by DNA polymerase, and then ligation. So all the same steps are going to happen. So here you have induction of double strand breaks by an endonuclease in the MAT alpha locus, that is known as HO endonuclease. Then there is a 5' to 3' dissection. Dissection means it is excision, right? So it is moving in 5' to 3' direction. Then there is strand invasion, right? Remember the steps that are involved in homologous recombination: strand invasion by RAD51 gene, and then there is new DNA synthesis at the homologous sampling. So new synthesis is possible because template is already there. It is homologous, so it can be refilled. And then so there is the corresponding long 3' tail at the bottom strand is displaced, and then the new 3' end is used to prime and extend the copy of the second strand. So this is a gene conversion event. It is known as gene conversion without crossing over. No crossing over is happening. No two different molecules are involved. They're not overlapping each other. They're not crossing over. It's known as gene conversion event. So gene conversion event is responsible for mating type switching in yeast without any crossing over. And it can help convert one mating type to another in body yeast. And this is because of the recombination which happens in the yeah. So even if you just understand this figure, what is happening, it's enough. And just understand that this is known as gene conversion event. This term should be clear, which is without crossover, right? So homologous recombination is happening, but without crossover. So we're done with this part, and we're done with homologous recombination, and now we're going to move on to site-specific recombination. So in site-specific recombination, We need to do conservative site-specific recombination and then transposition. So there are two processes as you see here, number 1 and number 2. So today we are going to do CSSR, which is conservative site-specific recombination, and in the next class we are going to do transposition. So these are the two small topics here. It's not a very big unit, right? So these are the classes of genetic recombination. It is similar to homologous recombination, and it also involves pairing of two homologous regions which come together, and then there is recombination. But it is little more precise as compared to homologous recombination. You could call it, it's a kind of sub-part of homologous recombination, where the regions of homology are very small. It is not a very extensive homology. The regions are very small, and the certain sites only where the recombination is going to happen. Whereas in a homologous recombination, for example, the homologous recombination that happens during meiosis, you have those complete chromosomes aligning with each other. The crossing over can happen anywhere, and so the recombinant gametes can be formed using any kind of permutation combinations. So you cannot define what are the regions that are going to recombine and what will be the outcome of this recombination. But here you precisely know what is the outcome of the recombination because it happens at precise sites known as recombination sites. It will not happen away from that. So those recombination sites only are the regions of homology. So it's a very short ranged phenomenon as compared to homologous recombination. It is site specific, very, very site specific, right? But otherwise the principles are the same. You will see the processes are also very similar, right? So it's a conservative site-specific recombination which happens between two specific sequence elements, and the examples which involve CSSR are insertion of viral genomes. You must have heard of lytic cycle and lysogenic cycle for virus, right? For phages. So phages like bacteriophages. Bacteriophages are viruses which attack bacteria, and then you have other viruses also. You must have heard of HIV. HIV can, remain dormant in your genome for several years, and then it can again be reactivated at some point of time. So why is that possible? How is that possible? That's because the genome of the HIV can integrate itself into the human genome. It can stay there silent for some time before some kind of stress condition arises, and it switches from its lytic stage to the lysogenic phase. Lytic phase is when it is dividing, replicating itself, making its viral particles, too many particles within one cell, so that the cell has to burst open to release them. And then these particles can attack the other adjacent cells. So the infection spreads. So that is known as lytic cycle, because there is lysis happening. So it is lytic. And the lysogenic is the one where the genome of the virus gets integrated within the host genome. It can stay there silent for a long time. It could be 1 year, it could be 2 years, it could be 10 years, it could be 2 decades. It could be few months, it could be few days, it could be anything, depending on the condition, the immunity of the organism, the kind of condition that he is facing, the kind of lifestyle the organism has. So it can be reactivated or presence of some other diseases alongside. So lysogenic cycle involves integration, and that is the site-specific recombination, which is the insertion of viral genomes into the DNA of the host cell during infection. The other example is the gene rearrangements. The gene rearrangements that happen that modify our genome. Why we are different from our precursor, or let's say your ancestors? Because even in humans you see a lot of evolution. We've come from Neanderthals to Homo sapiens to us, what we have now. Homo erectus. So there's so many different stages that we have crossed. Why? Because that evolution is still happening. So it is not just mutations that are happening, there is a lot of genomic rearrangements that happen. All those genomic rearrangements are possible because of site-specific recombination, which can result in things like inversion, deletion, duplication, and they involve bigger regions as compared to single point mutations. Because single point mutations are not going to change one genus into another genus. It can change one species into another one. For example, I would say that Homo erectus may have become Homo sapiens because of few mutations, but a chimpanzee becomes a human. It could not be possible just because of mutations. Had that been possible, we could have become chimpanzee anytime. No? It is still possible if single mutations can do it. So these are long genomic rearrangements, and they are because of site-specific recombination that can induce things like inversion, deletion, duplication, right? So these things are also because of the conservative site-specific recombination. We'll come to this reason why we call it conservative site-specific. Then the next phenomena is transposition. So this we are going to do in the next class. Transposition is also a kind of recombination, and it happens between specific sequences and non-specific sequences. So, like in homologous recombination, the site-specific recombination is also between specific sequences, which means both the regions are specific. You know where it is going to happen. If you look at the genome, you can specify. You can tell that this is the region where it is going to happen. In case of transposition, you cannot say anything. One region is specific, the other you do not know. And that is what transposons do. Transposons are jumping genes. So you know the sequence of the transposon, but where it is going to insert in the genome, that you don't have any idea. You can randomly go into any gene and make it become a pseudogene or a non-functional gene, or just make it not getting expressed if it goes in the promoter region or the enhancer region. So here one region is one site is specific, which is your transposon sequence, but where it is going to insert you do not know. So it is between a specific sequence and a non-specific sequence. Does it make sense? Right? Whereas in site-specific, both the sites are known to you. They are two specific sequence elements. You recognize both of them, and you precisely know here is the point where the recombination is going to be. You can predict the outcome of that recombination. Right? So the example of transposition is when you have movement of transposable elements, which is the major source of spontaneous mutation, and nearly half the human genome is derived from transposable elements. If you have heard of some elements like line elements, sign elements, have you heard? LINE, they're all capital. It's called long interspersed elements. So long L, interspersed IN, then elements E. So LINE, it's called line. So similarly, you have short interspersed elements, it's called sign, SINE. So lines and signs, all these are kind of transposable elements which are derived from transposons, and they are very much there in our genome. All that 97% genome which does not code is all made up of transposable elements, and they bring a lot of rearrangement. They bring a lot of they make a lot of genes pseudogenes. They make a lot of pseudogenes genes because they can just go in, they can just come out. They're jumping genes. They can just move here and there. Now coming to the two classes, I'm just showing you pictorially how does it happen, and then we will focus only on the site-specific recombination. So here we have these recombination sites. So they are defined that this is this red one and this is this blue one, and then we have some region which is a lighter one. So that is a region flanking. It is region which is flanked by the recombination sites. So this region is flanked by the recombination sites, this red and blue one, and now because there is a site-specific recombination between the two, which means they are homologous, so they can come together and then they can be a switching. Because of that, now the B moves here and the A moves here. How? Because this whole of this region is inverted. It moves like this. It is like this, it moves like this. It gets inverted. Right? There is cutting and then there is looping. Inversion. So that is why they have drawn A and B upside down. Like A and B are upright here, and here they're upside down. Right? So there is inversion. It is showing you the inversion. And so half part of the blue moves here, and half part of the red moves here. Right? And you can precisely make out what is going to happen. Even if it doesn't happen, you can say that this is going to happen, this is going to be the outcome. Because it is going to happen between these two sites only. And this is the region that is going to show you inversion because it is flanked by recombination. That defines it. So here recombination between red and blue recombination sites inverts the DNA segment carrying the A and B genes. So that is why A and B genes which are present at the boundary of this flanking region flanked by the recombination sites undergoes inversion. So this is an example of transposition. Now this is a transposable element. So this is a specific sequence, but where it is going to go we don't know. So this is some sequence we don't know, and it can go anywhere inside that. So this region now moves from here to here. So the gray thing remains here, and the blue thing gets incorporated. In the blue molecule, this red thing gets incorporated. Is this clear? So the red transposable element excises from the gray and inserts into the blue. Okay, so today we'll do conservative site-specific recombination, which is abbreviated as CSSR. Now, so this is recombination between two specific DNA sequences or specific DNA elements, and it is catalyzed by proteins because obviously you require something to put a cleavage and then to rejoin in a new way. That is what is recombination, right? So because inversion can only happen if you do cutting and then you do a flipping, right? So this is catalyzed by proteins known as recombinases. So recombinases are the enzymes that catalyze this process because they recognize these specific sequences. So the recombination sites that we saw here, these ones, they are recognized by the recombinase enzymes. We will also study the structure of those recombination sites, and they bring these sites together to form a protein DNA complex, bridging the DNA sites known as a synaptic complex. You know synapse is also there in case of neurons, where two neurons come together. There is a bridging where neurotransmitters are released, and there are receptors on the other neuron. So that is why the same name is given, synaptic complex. And even synapses is also a term which we use in meiosis during prophase stages also, right? Synaptoneural complex and all. So when these recombinases are making a cleavage within the recombination sites, then they form a protein DNA complex, which means this recombinase protein is going to now form a covalent bond with the DNA, and then there is rejoining. So within the synaptic complex, the recombinase catalyzes the cleavage and rejoining. So remember, these are the two steps. First is cleavage, second is rejoining. And what is the outcome? They can either invert a DNA segment or they can move a segment to a new site. Now, if there is inversion, then you know that the location remains the same, but the orientation becomes opposite. Like you had AB here, so now A here, B here, right? That is inversion. Whereas the other thing which can happen is it could be a movement of the DNA sequence from one region to another region. So what could that lead to? What kind of rearrangement is that when the location of a particular region in the genome changes from one location to another? Translocation. Transposition is not this. Transposition does not happen between the recombination sites. It requires only a transposon and a non-specific sequence, which we don't know. Right? So it is translocation. Translocation, that you know, the kind of genomic rearrangements where some part of the chromosome goes to another chromosome, or maybe within the same chromosome. So within the synaptic complex, both the things can happen. Now recombination sites. So recombination sites consist of two classes of sequence elements. They have sequences that are specifically bound by the recombinases, because recombinases are the one which recognize them, right? That is why recombination happens there. And the sequences where the DNA cleavage and rejoining occur, which is the activity of the recombinase. So you require sites which are recognized by recombinase, and you also require sites where cleavage and rejoining happens, so that the recombination can actually happen. So CSSR can generate three different types of genetic rearrangements, like insertion. Insertion means that because you're saying that one region gets excised out, it moves to another region. So if you look at the whole genome, you would call it a translocation. If you suppose you can make out that one chromosome is broken at some point and you see that region somewhere else, so that is translocation. But if I just look at one genome, look at just one chromosome, I would see, and I don't know where it has come from, I just see an extra region, so I would say something is inserted here. When you look at the bigger picture of the karyotype, it is like a translocation, if you can make out where it is coming from. If you cannot make out where it is coming from, right, then it is just an insertion. Do you get the point? And if it is nearby, you would even call it duplication, and if it is the same sequence. So you can have different contexts to say the same thing. If you look at whole genome, something moves from here and goes somewhere else, and you can see missing part here and you see extra part there, so it is translocation, right? But when I look at just one chromosome which is having something extra, you say there is an insertion, right? Now what is inserted, if it has a same copy somewhere else on the same chromosome, you call it duplication. So this is doubled because another copy is inserted. Does it make sense? You look at whole genome. Suppose you had this one chromosome and you saw this is truncated, and then you saw another genome and then this is elongated, and you can make out that this is what was here is now attached here. So it is translocation. If I just look at this, don't look at this, then what do I call it? Insertion, right? And suppose this region which is here is also here. Duplication, because I see the same thing again, right? So they're different contexts to say almost the same kind of genetic variant. Yeah. Transposition is not what we're doing today, so don't even think about it. We're not doing transposition at all, right? So that we'll do in the next class, you will understand. So insertion of a segment of DNA into a specific site, for example, even integration of bacteriophage genome or a viral genome in the host genome is a kind of insertion. We're inserting a viral genome into the host genome. That is also kind of insertion, right? We in terms of virus, we generally call it integration because it remains there for a very long time. It could be several years. It could be during the lifetime of the whole organism, right? So it is integration. Next is deletion, which means something gets excised out. And third is inversion. Inversion is what we did in the last slide, right? So these are the three kinds of genetic rearrangements. And we'll also come to figures where I show you all these. Now this is your integration of phage genome, and this is particular phage known as lambda bacteriophage, right? Lambda bacteriophage is one such bacteriophage which undergoes lysogenic cycle. It can switch from lytic stage to lysogenic stage depending on the conditions in the host genome, host cell, in the host cell. So the integration of the lambda genome into the chromosome of the host cell is what is shown here. So this bigger one shown here is your bacterial recombination site, and the smaller one shown is the phage genome. Although it is not to scale. Why it is not to scale? Because the phage genome is very, very small as compared to bacterial genome. Phage genome will be like few kbs, 5 kb, 10 kb. It could be even lesser than that. And bacterial genomes will be in the order of MBs, right? But what you see here is hardly 1/3 difference. So that is not there. It is of the order of 10^3 and even more than that, right? So that is why this is not to scale. Don't imagine that phage genome is this much in front of the bacterial genome. It is much, much smaller, but just to show you how it happens. So this is the phage genome and this is the bacterial genome, and they're together inside one cell, because you know that phages insert or inject their DNA into their tail fibers. They sit on the bacterial cell and they can inject their DNA into the bacterial cell, right? That is what viruses can do. They can inject their DNA into the bacterial cell. And now there is recombination between the two. And the recombination is happening at the recombination sites. So you see specific sequences here and you see specific sequences here, and the cleavage happens almost in the middle of these recombination sites. That is why at the end what you see is half blue, half red, half red, half blue in the whole thing. The whole of this phage thing is now inserted within the host gene. The host is a bacterial cell. Bacterial cell. A similar thing can happen for animal cell also. Right? So the cleavage happens within the recombination site. That is why we say that recombination sites have two kinds of DNA sequence elements. One, recognition site for the recombinase enzyme, and second, the site of cleavage and rejoin. Right? So that is what is happening. So recombination sites. So each recombination site is about 20 to 30 base pair, and it contains a pair of recombinase binding sites which are positioned symmetrically, and they serve as recognition sequences for recombinase. And they also have the cleavage and rejoining site, right? So the cleavage and the rejoining site. So so each recombination site contains this as well as this, right? Don't think that there are two types. No, each recombination site contains these two things. So they will have recombinase binding site as well as they will have the cleavage and rejoining site. And the recombinase binding site is actually positioned symmetrically. So it is generally in a pair. Because recombinase has multiple subunits, and so one subunit binds to each pair of the site, each pair of the recombinase binding site within the recombination site, right? This is the recombination site, and within that you have this recombinase binding site. Don't get confused between that. Okay? So in the DNA cleavage and rejoining site, you have the crossover region. Crossover region where the crossing over is going to happen or the strand exchange has to begin. So this is where the strand exchange occurs, and then there is a central short asymmetric sequence. So here you have symmetric sequences and here you have an asymmetric sequence. And the crossover region is the one that also defines a polarity. It gives a polarity to the recombination site, that which direction it is going to function in, 5' to 3' or 3' to 5'. So look at this molecule here. So you see this whole region that is flanked by gene A and B, right? So there's gene A here, gene B here, and the whole region flanked by gene A and B is basically your recombination site, where the pair of recombinase binding sites is the red one. So red on the left, red on the right is your recombinase binding site, and in the middle that you see this gray portion, that is your DNA cleavage and rejoining site, because that has a crossover region. And this crossover region has a polarity, that is why you see an arrow here. So it is going to function in this region, this direction. So one recombination site is going to pair with another recombination site with another molecule, because it is homologous recombination. Obviously you require two molecules to come together and then they will be combined. So when they come together, then there is a strand exchange between the two because of crossing over. Crossing over always happens in the region of this arrow, which is a crossover region, because that is the region where the cleavage and the rejoining has to happen, while the recombinase enzymes will bind on the red portions here or on the blue portions here. Okay, just read here also. The darker red and blue boxes represent the recombinase recognition sequences. Recognition means the recombinase binding sites or the recognition sequences, and the black arrows show the crossover regions. Okay, so there is crossing over between this region after the recombinase enzymes bind this region and this region, the dark red and the dark blue. Okay. So these are the structures involved in CSSR. So these are the recombination sites, and it can switch to this form soon after recombination, which means when these two come together, then the blue moves up, the red moves down. So it becomes red, blue, blue, red. Similarly, this blue can also move up and red can come down, so it can become blue, red, red, blue. Either way. Okay, so there is recombination. So these are the sites of recombinase recognition, while the middle one, this arrow containing one, is the one which is for the crossover. So now it is showing you an expanded view of the same thing. So the recombinase recognition sequences are here, and they are symmetrically placed around the crossover region. The crossover region is here, right? So they flank the crossover region where the recombination is actually going to occur, because recombination is going to occur by cleavage and rejoining, and cleavage and rejoining occur within the crossover region and not in the recombinase recognition sequence, which is the red, red and blue. It happens in the gray portion, middle one. The subunits of the recombinase bind these recognition sites. Okay, and the sequence of the crossover region is not palindromic. You know what is palindromic sequence? What is read same from either side. When I read a sequence from 5' end or from the 3' end, it is same, then it is palindromic. For example, you have the restriction enzyme sites which are palindromic, like GAATTC. If you write a complementary sequence and again go in the same direction, So it is 5 dash, it is 3 dash, 3 dash, 5 dash. Read the above sequence in 5 dash direction. It is GAATTC, and when you go from this direction, 5 dash again, GAATTC. So this is palindromic sequence. So these are not palindromic sequence. Look at the sequence in the middle. It is TAGC, but when you read it from the other end, it is GCTA. So they're not palindromic. So the crossover sites look symmetric, but they're actually not. They're not palindromic. So the sequence of the crossover region is not palindromic, resulting in an intrinsic asymmetry to the recombination sites. That is why there is a polarity. And it's been same from both the sides. You go this way, GATTC. You go this way, GATTC. There is no polarity. You can read the same sequence from either end. But here there is a polarity, which means when you go from this direction, it is different. When you go from this direction, it is different. Right? And crossover happens only in one direction here, which means that this is recognized in this direction only. So if it is right, right. If it is left, left. 5 dash to 3 dash or 3 dash to 5 dash. So that is why you have an asymmetry here, whereas the recombinase recognition sites are placed symmetrically, and they could be direct repeats or they could be inverted repeats. Inverted means this sequence and this sequence is almost similar. It goes like this and like this. Opposite orientations, but they could be similar, right? And so there will be homology between this and this also. That is why there will be homology between within the crossover region. That is why cleavage and rejoining is going to happen there. Homologous recombination is going to happen there, right? So you have TAGC here, you have TAGC here, you have ATCG. So in the crossover regions, the homology has to be there. Then only the crossover is going to happen. That is why it is coming together, right? Whereas these sites are going to only function for the binding sites for the recombinase enzyme to come, so that it can catalyze the cleavage and rejoining within this crossover. Okay, recombination is not going to happen in these blue and red sites. That is going to happen only within the homologous region, which is the crossover region. So these are the molecules of recombinase. So we see 4 molecules that are bound here, 2 red, 2 blue, and then recombination is going to happen. What's the time? So just give me 1 second to complete this. So you have this sequence here, which is against this. There is recombination happening here, and it can result in insertion. So whole of this red thing inserts into the blue thing. Here, there is a looping because within the same molecule you have the recombination sites, so they can come together the way it was happening in yeast, mid-time switching. And now it can result in deletion. So the whole red portion deletes out. And here it can just form a U-shaped molecule like this, and it can result in inversion. So the C and D become inverted. So it can result in insertion, deletion, and the inversion. Right? The first entity. These are 3 kind of generations. Yeah. So those who want to leave for the class.